Hello everyone, in this video we are going to talk about what?
What we are going to talk about? I'm going to talk about is it what? Is it a correct way to use we in every video to announce anything like B or I? It could be it could be what? It could be what? I think video is recording fine. Is there any problem in it? Yeah, it's correct. Okay, we are talking about. Okay, how do I going to pronounce it? Let me search about it. My camera is not. Yeah. I think it's fine. It's fine now. Mm. Are you able to hear me? Yeah. No. Are you able to watch me? Or you can, or you can, or you can, or you can see me? Let me adjust again it. Shoulder. Yeah. 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 Do it again. Now I think it's correct. Okay. Hmm. Hmm. Let me adjust it again after this video. Now focus on what? How to pronounce CRISPR or CRISPR. CRISPR. How to pronounce CRISPR? What is it? Cas9. CRISPR brief introduction. English. Yeah. This is 9 in English. CRISPR-Cas9. Yeah. Yeah, I also think about that like CRISPR-Cas9. But there are what like UNESCO, UNESCO used to describe it in a single word. UNESCO, UNESCO, WHO, WHO. We use W each word separately in order to pronounce that word. Like good is not. If we pronounce good, we use all word together in order to pronounce that. But in case of WHO, we use single word separately in order to pronounce it. That's why I was thinking about this word. So no confusion is got clear. It's going to pronounce like what? It's going to be pronounced like it's going to pronounce like CRISPR-Cas9. So Cas9. Cas9 is a revolutionary gene editing technology that allows scientists to precisely modify DNA with a living organism. The acronym CRISPR stands for the clustered regularly interspaced short palindromic repeats, which are specific sequences of DNA found in the genomes of bacteria and archaea. These sequences, which were originally part of the bacterial defense system against viruses, have been adapted for use in genetic engineering. Genetic engineering is a revolutionary gene editing technology that allows scientists to precisely modify DNA within living organism. Precisely modify DNA within living organism. Oh, is it possible? We can modified DNA within living organism. The acronym CRISPR stands for the regularly interspecies short palindromic repeats, which are specific sequences of DNA found in the genomes of bacteria and archaea. These sequences were originally part of the bacterial defense system against viruses, have been adapted for use of genetic engineering. Wow, it's going to be quite interesting to read it out. So let's move to the next slide of this presentation or concept slide. CRISPR-Cas9 system consists of two main components. CRISPR sequence of the genome of the organism contains short repetitive DNA sequences interspersed and unique sequences derived from viruses and other foreign genetic material. These unique sequences called speckers serve as a memory of past infection. When a virus invades an organism again, the CRISPR system can recognize and target the viral DNA using the spacers as a guide. Cas9 is an enzyme that acts as a molecular scissor. It can be programmed to cut DNA in a specific location guided by a small piece of RNA known as a guide RNA or gRNA that is designed to match the DNA sequence of interest. The gRNA direct Cas9 to be the precise location in the genomes where the desired genetic modification to be made. Okay, we are read about it. What the case? What are the meaning of the different things? Like CRISPR and Cas9. Now back to the other thing. The workflow of CRISPR-Cas9 gene editing technically involve the following step. How the genetic editing process began, all things have described in this step. First, designing the gRNA. Okay, scientists design a custom gRNA that matches the target DNA sequences that want to edit. Design a custom gRNA that matches the target DNA sequences that want to edit. The gRNA will guide the Cas9 enzyme to a specific location. Deliver the gRNA and Cas9 are introduced into the target cell. This can be done using various delivery methods such as viral vectors, lactoportion, and nanoparticles, depending on the type of cells and organism being modified. Cas9 cleavage. Once inside the target cell, the Cas9 enzyme guided by gRNA locate and cleaves the DNA on the desired site. They create a double stranded break in the DNA repair. The cell natural DNA repair machinery then comes into play. There is a primary repair path, non-homologous end joining, and homologous directed repair. NHEA. This is error prone and can result in the intersection and deletion at the cut site, potentially disrupting and gene function. HDR is a more precise repair mechanism that can be used in the insert specific DNA sequences at the cut site. CRISPR-Cas9 technology has a numerous application, including basic research, gene editing, engineering or organism development of disease model, and potential therapeutic intervention of genetic or disorder. Its simplicity, efficiency, and versatility have made it a game changer in the field of the molecular biology genetic. However, it has also raises ethical and safety concerns, particularly in the context of the human germline editing and unintended off-target effect. Okay, we will read about what the workflow is. First, we design a GRN based on the DNA of the particular organism we are going to implement it or going to edit it. Then delivery, then GRN can introduced in the target cell. Then they introduce those GRN in the target cell. That can be done using various methods like vector, or the various method can be used for that. Cas9 cleavage. Once inside the target, the Cas9 enzyme guided by GRN located cleavage the DNA on this side. This created all the standard criteria. They break down the DNA in the different method. DNA repair, the neutral DNA repair machinery then come into play. There are two primary repair pathway: non-homologous and joining and homologous direct repair. These are the two different type of method can be used in order to repair it. CRISPR technology numerous applications. There are various numerical and genetic engineering, organism development of disease model, and potential therapeutic intervention of genetic disorder. Its simplicity, efficiency, and versatility have made it a game changer in the field of the molecular biology and genetic. These are the things that we have discussed about genetic. Genetic what the how the workflow is, how the workflow, how what's the workflow of in order to implement a grna, then there is a report of DNA repair. All this kind of thing happen in order to work of the CRISPR-Cas9 gene editing. These are some questions that we are going to discuss, and I think this is the last slide of this presentation. There is no other question. How is a CRISPR-Cas9 is in a biological research? How is a CRISPR-Cas9 used in a biomedical research? CRISPR-Cas9 is used in biomedical research to study gene editing, create disease model, and develop potential treatment for genetic disorder. Yeah, it's used in different field. It's used in a different field, as we can so the CRISPR-Cas9 is used in biomedical research to study gene editing. It's used in a different way, like gene function, create a disease model, and develop potential treatment for genetic disorder. What role does CRISPR-Cas9 play in agriculture? In agriculture, CRISPR can use to create genetically modified crops with desired traits such as the pest resistance or improved nutritional content. Okay. This can be used in the field of agriculture also. That's a magnificent achievement. Okay, are we move to the next hard point of this slide? Okay, yeah. Should we move? Are you are you listening or thinking about something else? Too patient. Do listen here. Ah, bro. When I'm going to when I will become fluency, when I will achieve fluency in that skill, I am giving whole focus from last month. Not for that period, I'm not that focused, but from last month of 15 or I think 16 August, 15 or 16 August of 2020, I'm quite focused on it. Already I've uploaded about what 160 videos, 30 video are all are about previous 30 video are all about previous videos. 30 videos are previous, and and what and what and what uh, but in the last one month I already upload around 130 videos. Now at a spend period of time, it's a quite big achievement for me. It's a quite big achievement for me. And after 200 video, I'm thinking about to change the format, where the slide you can see on the screen also going to be write down my itself, because it's going to help me in writing skill as well as speaking skill. Not just understand us, what in order to improve your speaking English, to understand writing skills also. That's one of the more big factor of what speaking English in order to achieve an fluency in it. So from today or tomorrow, not tomorrow, but when we achieve 200 videos, when I will achieve 200 videos on this channel, and I'm going to change the format and keep going with it, because this is the ultimately going to help my fluency. Like, I'm going to speak it about what grammatical mistake, 20 question of grammatical mistake, all this stuff. There are going to be 5 video at a day. Those 5 videos will be different from each other. That is going to help out. Now to the video, to the slide. What are the significance of the CRISPR-Cas9 in biotechnology? How to CRISPR can contribute to the disease diagnosis and detection? What are the some environmental application of CRISPR-Cas9? In what way does CRISPR-Cas9 impact the study of the evolution and ecology? These are the some questions that given you can read the answer of them. What the potential role of the CRISPR-Cas9 in the organ transplantation? CRISPR-Cas9 may be used in a modified donor organism to make them more compatible and replicant, and potentially reducing the risk of the rejection in the organ transplantation. How the CRISPR-Cas9 will be applied in the conversation of endangered species. That's it for the video. We'll see you next time.